And thank you all for coming. It was really nice to meet you last week. And we'll be there. I'll be there next week. We have a dog problem and Patty can't come next week because of it. Too many dogs. But in any event, what I want to start out with is a fitting of the Paint Rock project into this global Earth Observatory thing at the Smithsonian called the Forest Global Earth Observatory. It's the successor to something called the Center for Tropical Forest Science because it was needed to be changed because we now have representations of forests in boreal areas as well as in the tropics and temperate zones as well. And Paint Rock is one of the temperate sites that we're going to add. Here is a map without Paint Rock of the 71 plots that we have around the world now. You can see they're mostly in the New World and in China. There are some appearing in Africa, but none yet in Australia, and South America is somewhat underrepresented. But nevertheless, we have 71 plots in 27 countries. And the personnel that work in these plots are representative of the diversity of humanity. I mean, it is an amazing collection of people. We have more than 100 partnering institutions, some of whom are banks and things like that that give us money. There are 6.4 million trees with a tag on them. More than about 12,400 species, which is estimated to be about a quarter of all the trees in the tropics at least. It's probably 20% of the trees in the world that we have a representative in the plots. And so far we have about 18 million measurements that we've taken. So you guys join a pretty large effort that's going on, and it will be really valued as we go forward. So here is the place where the first plot was established, an aerial view of Bar Colorado Island. It's in an artificial lake, Lake Gatun, that was created to provide the water supply to run the locks. And so when it was, it's a former hilltop that got flooded and isolated from the, quote, mainland, unquote. It's 15 square kilometers. There's a big boat going through the canal. So we started this in 1980, and it took us 2 years to do the first census of the 50-hectare plot. But we've now done 8 censuses. We were going to do one last year, but COVID put a kink in that plan. So here's the trail map for BCI. The 50-hectare plot is right here on the summit of the island. This is Panama. It's an east-west country for those of you who are geographically challenged, as I was. It's weird to get up in the morning and find the sun coming up in the west. It takes some getting used to. Half of Bar Colorado Island, the hatched area here, as far as we know, has never been cut. It's not perfectly virgin. There may have been a few mahogany trees taken out at one point in the last century, early in the last century, but we don't know that. And this is 1 kilometer by half a kilometer. So this is smaller actually than Paint Rock. This side of the island was in farms and has regenerated for the last 120 years. So it looks pretty old growthy itself, but we wanted to establish it pretty much in uncut forest. So here's the lab complex at Bar Colorado Island. It's quite a fancy place to live. I call it a dude ranch for biologists. It's a wonderful place. And over here, we have some examples of maps of individual species superimposed on the topographic map of the plot. By the way, these first slides were provided to me by the director of the Global Forest Network, Stuart Davies, and so I'm grateful to him. So we have a lot of large plots. They range in size from 16 to 150 hectares. Sorry guys, 60 doesn't quite make the grandest size, but it's one of the largest plots we have. We map, measure, tag, and identify all trees over 1 centimeter. So this should be familiar to all of you. That's exactly what we do. So there's one of the things that makes this a wonderful data set is the methodology is totally standardized. So everybody can be sure when they're analyzing the data that they have the same kinds of measurements. All the trees get measured in the network every 5 years. I'm not sure what's going to happen at Paint Rock because of the challenges of funding it, but possibly we'll do a second census according to Dawn within a 3-year period. And after that, we may go to a 5-year rotation. We also have some measurements of tree growth and things and mortality every year. And then we take a lot of measurements physically. There are climate stations at most of these sites. And we've done soil chemistry at a lot of the sites and so forth. So here's a piece of real estate that most of you from Alabama A&M know well. And this is a 50-hectare rectangle superimposed on your campus, just to give you a sense of how big these things are. Here's a football stadium, a small area within the 50-hectare plot. Of course, at Paint Rock, we have another 10 hectares out here that we want to add. So we'll have a really large sample. So why do we need these huge plots mapped, and why do they need to be long-term? Well, to understand the dynamics of a complete forest, you have to study all the trees simultaneously. So it doesn't do any good just to tag the individuals of one tree species. You've got to know what all the others are doing, and you've got to know how they interact with each other. Secondly, most of the trees are uncommon to rare, especially in the tropics, but surprisingly not untrue of Paint Rock too. So we've got to have large sample sizes to get enough individuals to do population level studies to analyze. And the other thing to say is that most of the theories to explain how tropical forests or temperate forests work are explicitly spatial. So we need a map of the individual trees. We have to know where they're growing next to one another. Trees are rooted organisms, and so their interactions are mediated by space. And the other problem, of course, is that trees, they're not a good thing to study if you're an old man like me, but trees live a long time. And so this requires long-term studies to get the dynamic information. And finally, you can't just look at the big trees. You've got to also look at the small ones, the saplings. And the reason for that, that'd be great. The reason for that is that most of the dynamics that determine the fate of populations happen in the young stages. So here's another slide from Stuart showing the sort of stages of work. First, you survey the plot with a theodolite, equipped with a laser nowadays, not in the old days. Then you map and record all the trees. You collect the specimens and make a herbarium, identify them. And you have to hire a lot of people. And here's a site. This is from Lambir in Borneo, in Kalimantan. And the crews are very large and very diverse in race, which we are very proud of. And each of these sites is often remote, and so we have to have field stations that we run and operate and hire cooks and all sorts of things. You guys have it easy compared to some of these sites. And this is a graph from Robert Whitaker's community ecology book showing a famous graph of the relationship of biomes to annual precipitation on the y-axis here and annual temperature on the x-axis. You can see the sampling ranges over most of the forested biomes that we have on the Earth, but there's still some clear areas where we're short, and the temperate zone is one of those areas where we need more studies. But my grail is understanding tropical diversity and rarity. And these plots are useful for huge numbers of questions like carbon sequestration and responses to climate change. But I want to know why there are so many tree species in these forests and why so many of them are extremely rare. And so I want to just emphasize these 2 points in the rest of the talk and try to get some discussion of this going about what's going on maybe at Paint Rock later on. But here's a fact that is pretty shocking to first timers. If you count up the trees on Barr, Colorado Island, which is a few square kilometers in size, it has half as many tree species as all of North America north of Mexico, an area that's 44 million times larger. What the heck's going on? Can we explain the maintenance of all those tree species in such a small area? Well, not fully yet. That's my grail, but we're making some progress, and that's what I want to talk about in the rest of the talk. So I thought it might be more useful for me to show you the results from the longest running of the forest geo plots, the one on BCI since 1980. Some of the things we've learned about diversity and rarity in this forest over that time frame, just to give you a sense of where we're headed with the Paint Rock study if it goes on as long as we hope. So I want to focus on the role of chance and determinism in the dynamics of the neotropical forest, that is the one on BCI. So the organizing question, and one of the fundamental ones in community ecology, is a pretty big question. To what extent are the dynamics of ecological communities dominated by non-stabilizing stochastic forces, that is chance effects and things like drift, versus stabilizing deterministic forces such as density and frequency dependence arising from the adaptive differences among species, that is niche differentiation or things like predator-prey interactions or host-pathogen interactions. And what we're going to see in the rest of this talk is that both of these, it's not an either or, it's a mixture of both of these things going on in BCI at least. So what we're trying to do is test for evidence for stabilization in the population growth of tree species. On the left, we have an unrestrained population growth growing exponentially. And the characteristic number for this kind of a curve is something called little r, or the intrinsic rate of increase. It's basically the logarithmic slope of this line. But when we're looking for evidence that stabilization is maintaining populations in the forest, we look for all species having some sort of curve like this, where they stop growing fast and decline in growth rate until they reach an upper limit. And that limit is called the carrying capacity. And there can be all sorts of reasons for what sets this number, but what we're first trying to figure out is how many species have this kind of growth. Now, I got into a lot of trouble with my colleagues 21 years ago by publishing, or 20 years ago, by publishing this book called The Unified Neutral Theory of Biodiversity and Biogeography, which argued that ecology hadn't really done a good job of testing the role of chance in community ecology. And in particular, I said, you know, like population genetics, we can have drift in communities. We can have random changes in abundance of species that are just due to differences by chance in births and death rates. And this should be especially important in very rare species. Now, this theory, of course, got me in trouble, and lots of people said it's not realistic. And I accept that fact, but it's got a life of its own. This book has now been cited 9,800 times. And this was a paper we published in Tree looking at the state of neutral theory 10 years after publication. I'm trying to get a group of us to write a 20-year retrospective, but we're not there yet. But I just want to show you one example of a remarkable fit of neutral theory to the data on relative tree species abundance across over 700 plots in all over Amazonia. And what this graph is a classical so-called rank abundance curve. And on the x-axis, we plot species rank in abundance. On the left, the most abundant species, and on the right, the least abundant. And on the y-axis, you take the logarithm of the abundance of the species. So it goes 1, 10, 100, 1,000, 10,000. And this is the curve that you get for the Amazon. And there's a number that comes out of this theory called Fisher's alpha, which is basically controls the shape of this curve. And I want to show you the fit of neutral theory that estimated the number to be 754 for the Amazon. And here's the fit. And, you know, that doesn't happen very often in ecology. I'm sorry. We're lucky if we get an R squared bigger than 0.5. And this is 0.9997. So it even fits the really rare ones. These are species represented by a single individual in all of the Amazon plots. Now, I haven't done the same thing yet for paint rock, but it's starting to look pretty good. And this is the graph we have, the rank abundance curve for the first year of data that all you guys, your predecessors collected, log abundance over here, and the abundance of species here. And this is a distribution that's almost what's called a power law. And it's going to have an alpha. I've forgotten, I think Rick Condit, one of our collaborators, has estimated it. And I don't remember the number that he came up with. It was 12 or 14 or something like that. But anyway, it fits pretty well. So I'm not ready to throw out chance as a driving mechanism for a lot of this diversity, but we have this huge number, even in paint rock, of very, very rare species, you know? That's got to be explained. So I want to dwell on this rarity a little more about tropical forests. So of the more than 12,000 tropical tree species in the network, the rarest half, that is about 6,200 species, add up to just 2% of the individuals in the whole network. Half of the species. Now these are really in trouble from a conservation point of view. Over 6,000 or 600 rather species are so rare that they occur as just a single individual among the 6.4 million tagged trees in the plot network. And we don't yet know if these locally rare trees are rare everywhere in the world, however. But this idea that tropical trees are rare goes back to Alfred Russel Wallace, who commented on that. He traveled widely in Malaysia and Southeast Asia. And he said it was very hard to find a second individual of the same species. So what's going on here? It's not explained very well. I mean, neutral theory is one explanation, but you know, it's got to be more complicated than that. Let me just emphasize this for one plot, which is the Barrow, Colorado Island plot. If you look at species ranked by abundance again on the x-axis and the cumulative number of individuals on the y-axis up to a percentage of 100, the top 9 make up half of all the individuals in the BCI forest, which has 323 species. Now, it's a little more than when this graph was made. But look at the rarest half of the species, 153 species, make up just 0.6% of the individuals. 0.6%, that's half the diversity. So what's the explanation? Well, the classical explanation is one that we heard from Drew. And I'm not saying it isn't true in those birds and true for a lot of rare species, but the classical explanation is that rare species are rare precisely because they're specialized for environments or resources that they require, but they can't, their requirements are not often met. The neutral explanation, of course, is that they just had bad luck. That is demographic stochasticity. Well, there's a newer explanation, at least for Barra Colorado Island, that rare species are more vulnerable to enemies, particularly pathogens, than are the common species. And we call this the enemy susceptibility hypothesis. And at the end of the talk, I'll give you the evidence that we're accumulating on that idea. So what are the conclusions? These are the three, I want you to learn these 3 points because they're the central points of my talk. So the first is that the 39-year longitudinal study since we completed the first census in 1982 has revealed a forest far from equilibrium, undergoing rapid non-successional compositional change despite strong conservatism in functional traits. Functional traits are things like wood density and leaf characteristics of chemistry and so forth and so on. Secondly, the BCI tree community shows evidence of both demographic stochasticity as well as deterministic environmental variance. And there is mounting evidence that the enemy susceptibility hypothesis may explain much of the rarity in tropical trees, at least in the BCI forest. And this is problematic for conservation because this mechanism of greater attack by pathogens of rare species is a destabilizing mechanism for diversity. So let's go back and look at these one by one. So the first is that the forest is far from equilibrium and that traits don't seem to explain the dynamics. So here's the evidence that we have so far. In the 39 years since the first census was completed, 40% of the tree species have changed in absolute abundance. These are trees now, by more than 50%. And 2/3 of the species have changed by more than 25%. Now, I don't know whether this is going to be true of paint rock, but this was really shocking to us. Now, this isn't true for every tropical forest in the network. It tends to be truer for forests that are on richer, more nutrient-rich soils, but it's still pretty amazing. And these changes occurred in species of all growth forms, from shrubs to trees, in species of all life histories, shade tolerant and shade intolerant species, and all abundances, common to rare. And these changes are not explained by functional traits among the BCI tree species. The community distribution of traits is nearly invariant. So we have a phylogeny due to John Kress and a former student of mine, Andy Jones, and others. And so are the changes in abundance explained by their phylogenetic relationships? And the answer is no. This was a paper published by Nate Swenson and others. They looked at phylogenetic turnover in the BCI plot through 2005 and functional turnover in the plot, functional trait turnover. Blue means changes were less than expected by chance, and red means changes were more than expected by chance. And what you see is that for phylogeny, the curve is symmetrical around a white bar, which is no change. So there's no net directionality in the change phylogenetically in the composition of the BCI forest. However, for functional turnover, the forest has more or less stayed conservative. That is, the traits have not changed. And if you look at the individual traits that they examined here and look at the different censuses, you see that the curves are essentially superimposed perfectly. So there's no change whatsoever. So I conclude from this that given all the change going on, having no change in the traits is not the source of the explanation. Now, Rick Condon has pointed out to me that these traits may not be the important ones that determine which trees are increasing and decreasing but they're the ones that were most convenient to measure and have been measured to this point. So what's the evidence that the BCI community has both demographic stochasticity or chance variation and deterministic environmental variance? Well, let me define my terms so that I can explain this a little better. Demographic variance or demographic stochasticity, I use that term to describe any population, any change in population abundance due to chance differences among individuals in birth and death rates. So sometimes births exceed deaths and the population grows, and sometimes deaths exceed births and the population declines. So I use the term environmental variance to describe changes in population abundance caused by all other non-stationary physical and biological processes that cause, and here's the important point, correlations among individuals in birth and death rates. So everybody's subjected to environmental variance and they tend to work in the same direction, but demographic stochasticity is random with respect to abundance. So demographic stochasticity should be more important in small populations, just as genetic drift is more important in small populations in population genetics. So if we go back to the graph of abundance by cumulative number of individuals, these are the species up here. The bottom rarest are the ones that should show more evidence for demographic stochasticity, whereas the common species should be more resistant to that kind of variation. And our measure is the measure of the population growth rate, which is the little r number I was describing before, and we calculated this for each census interval and each species, and it's defined as the log of the ratio of the abundance at time t, j plus 1 divided by the number at the previous time, j, divided by the time interval over which you measure the intrinsic rate. So this is a convenient measure of variance in population as well. And the nice thing is we can use it to differentiate demographic stochasticity from deterministic environmental variance. And in their book on stochastic population dynamics, Engen and Landy showed that demographic variance in species abundance can be measured by the absolute value of little r and how it changes with abundance of species. So it should decline as a power law with a slope of -0.5, but environmental variance should have no relationship to population abundance. And that's very nicely displayed on this graph. Here's the mixture of demographic and environmental stochasticity in the BCI tree community. If we had pure environmental stochasticity, the z-value of the slope should be 0, so it would be way over here. Conversely, if we had pure demographic stochasticity, meaning chance variation, we'd be at a slope of -0.5. And here are the values averaged across species and census intervals, so 1 through 6. And you can see that they're more or less 2/3 of the way to the stochastic component. This is largely driven by stochasticity in the rarer species, but one of them is more deterministic. That was the first one, and we had a very severe El Nino drought that killed 15% of the canopy trees, and that showed a big signal here in reducing the slope of change in R with population abundance. And if you look at the distribution of intrinsic rate of increase over BCI tree species, it more or less follows the neutral expectation, although there's a concentration of neutrality that we don't see in the empirical data, which are the black dots. So they are concentrated around no change, but there's some determinism here in populations that are increasing in abundance. But the rare end, the loss is more or less just what you'd expect from neutrality. So what I'm telling you is essentially, this is what we see out there. This is not what we see out there. We have no evidence for stabilizing carrying capacities to date. Now that may not be true later on, but for 40 years, this is what we see over here, not here. And another way to kind of illustrate that is that you would expect temporal decay in community similarity to be more or less continuous if neutrality is applied. And so there's a very simple way to display this. Imagine you have the abundance of any species at time t, and then you examine the abundance of the same species at time t+1, a short lag. You would expect a high correlation between those two because there's not been much time between the two measures of abundance. So you would get a high correlation coefficient or high coefficient of determination, R squared. But as time goes on and you have longer and longer lags, you expect this correlation to weaken and the oval to increase in cross-sectional area. And so you'd have a lower coefficient of determination. So what do we actually see on BCI? Well, here's the neutral explanation or expectation rather. Here's the lag in years that we have between the censuses. Here's the mean coefficient of determination. So when you do a self-correlation, of course, you have 1, but then we start to decay this fast. Well, how fast does the forest actually decay? One of the things we want to look at is to see whether it's linear, but whether it's superimposed on the neutral expectation. Well, here's what actually happened. It was steeper, but it's linear with time. So what this says is this is a mixture of neutral drift and deterministic processes that are driving a reduction in decay in community similarity with time. So it turns out if you run this out ad infinitum, the neutral expectation eventually reaches a lower plateau because it comes into equilibrium with species immigrating into the plot and those that are going locally extinct. Whereas we don't know what the observed curve will do. But it doesn't reach this plateau for several hundred years, on the order of 300 to 500 years. So there's been a lot of research over the years in the search for density dependence in tropical trees, and the most famous of these mechanisms is one named after Dan Jansen and Joe Connell, called the Jansen-Connell effect. And this is important because we need to find density and frequency dependence if this forest is in equilibrium and diversity regulated. If it's not, of course, and I've already shown you evidence that it doesn't appear to be, but we can look at the evidence for Jansen-Connell effects. And the reason we expect these effects is that these are rooted plant communities. So density dependence leaves a spatial signature, which is what Jansen and Connell talked about, that we can measure. So the basic model of density dependence by the Jansen-Connell effect is this. Here's a mother tree that's shedding seeds, and here's the dispersal kernel for the seed fall, which declines with distance from the mother tree. But if there are pathogens that kill seeds and seedlings or predators that attack them where they're most abundant, then no seedling will germinate or survive close to the mother, but the probability of survival goes up with distance. So if you take the product of how many seeds get there times their fraction that live, where the next trees recruit is at some non-zero distance away from the mother tree. And so this opens up habitat for other species to coexist with this particular species because it's not able to self-regenerate in the same space. And here's the evidence for Jansen-Connell effects from BCI. Basically, 75% of the tree species have zero recruitment immediately under the crown of the mother tree. But there are a small number of species that can recruit somewhat near the under the parent, but it's never more than about 15%. So it's always low. So What about other aspects of density dependence? Well, we might look at survival and growth rate. Here's a graph that looks at growth reduction for saplings that do manage to survive close to a maternal parent or a large adult at least. So this is the percent reduction in growth rate as a function of the size of the focal tree in dBH, diameter of breast height. So bigger trees have little effect. This is the line of no effect up here. And as you go further and further away from the mother tree or the large adult, the effect of that adult gets less and less. And so this would be no effect. But for focal individual saplings that are within 5 meters of a large adult of the same species, they have a rather large reduction in growth rate. But this effect diminishes the further you go away from trees. And in fact, the effect is very local. In fact, it only goes about 20 meters from any large adult. So that raises a question, is the local Jansen-Connell effect, which we can detect, regulating the abundance of populations of reproductive adults on the landscape? The importance here is that of reproductive adults. That is, if the density dependence we see doesn't regulate how many adults are in the population, it can't control that population's abundance. And here's a problem. It turns out that to date, almost all of the people who've measured density dependence among the progeny of single mother trees have not differentiated whether those seeds came from the mother tree herself or from another tree somewhere else. So it's like saying, if I kill off 99% of the seeds that land on their mother, but one of them survives to adulthood, that doesn't regulate how many mother trees there were. So this density dependence would not be regulatory. And knowing whether those seeds came from the mother tree or some other mother tree is simply unknown for most species. So what we did was take a different approach and say, is there evidence among the tree community for frequency dependence that is rare species advantage in the BCI tree community at the whole plot scale? If there isn't, this is not regulating diversity in the forest. And this is really important because frequency dependence that is rare species advantage is the expected community level consequence of all this negative intraspecific density dependence like Jansen-Connell effects. And the answer is here. Here's species abundance in 1982, and here's the last census we completed, the percentage in 2010. Sorry, there's 2015. It's not on this graph, but it shows the same thing. Basically, there's more variance in the survival rates for rare species, but this is just one standard deviation. And so these excess, okay, let me explain this. Replacement would be being above the line of 1 here, because this is the ratio of birth rates to death rates. So if this is 1, they're just replacing each other. And what this shows is that there aren't significant birth rates in excess of death rates when species are rare than when they're common. So we can't find frequency dependence at the whole plot scale. So this really raises questions about whether this forest is in equilibrium and density regulated. So after 4 decades, in other words, frequency dependence, rare species advantage, can be detected at small spatial scales, less than a hectare, but not on the scale of the whole 50-hectare plot. So these results have a very interesting implication, and that is that there is spatial scaling in the strength of density and frequency dependence. They are strong at very small spatial scales, but weak to non-existent on large spatial scales. I didn't mention this, but earlier, the fit to the rank abundance curve of neutrality assumes that there's no density dependence. And certainly on the scale of the Amazon basin, this is likely to be true, because what you're saying, if there were, is that fluctuations in the abundance of a tree, say in the Western Amazon in Peru, would somehow influence the population growth rate of the same species near Recife on the Atlantic Ocean, 2,000 miles away. So that seems very unlikely. So there has to be spatial scaling in density and frequency dependence, but as far as I can determine in the literature, there is no discussion of this scaling phenomenon in population regulation literature. So maybe there's an alternative explanation. Maybe the lack of frequency dependence is because all these tree species are at or near their carrying capacities. Remember, we're looking for stabilization. So if they're just fluctuating randomly around their carrying capacities, that would work. But the answer is the dynamics of the tree species themselves are inconsistent with this alternative explanation. That is, the tree community is very far from equilibrium, and I want to give you some graphical evidence of that. To study spatial population dynamics, what we did was compute the little r of each species in the 200 quarter hectare plots across the 50 hectare plot in total at each census interval. And fine scale Jansen-Connell dynamics are expected to produce a very homogeneous population response in spatial terms, but this is not what we see. And so what I've done is make a series of short movies of the spatial dynamics of a few species just to show you what happens. And what I'm going to show you is true of all the species we've looked at, which is most of them. So here's a species of understory tree, Desmonathus panorensis. And let me explain this graph. Here are the little squares are quarter hectares. Red squares are quarter hectares in which the population of the species is growing very rapidly at the beginning of our study near 1980. And blue squares are where the population is declining in abundance. And yellow and light green squares are where the population is more or less not changing. And so what I'm going to do is interpolate the dynamics of this species for the last 40 years, and you can watch how chaotic the dynamics is. And I challenge you to tell me of any population data you know that looks like this. So what you'll see is initially that there was, because of the early drought, the El Nino drought, we opened up the understory to light. And so this species responded by large scale population increases, but it doesn't persist. So watch what happens. Now, one of the things we get out of a study like paint rock is to compare the dynamics of BCI with data like this for a series of species. I mean, it's truly amazing to me. I've never seen anything like this. Here is another species. Oh, sorry, this is not another species. Here's another species. This is Hybanthus. This is the most common species in the plot. It has 40,000 individuals. It's an understory species, gets about 10 feet high. And again, it also shows the effect of that canopy disturbance with the death of the early canopy trees. But this goes through a massive population decline, much worse than the previous species, Desmonathus. However, at the end, it starts to come back. And we don't know if it's going to recover its original abundance or not, but maybe it will. We'll see. Okay, this is interesting because it is a Cecropia species, which is one of our gap pioneer species, and it has kind of a pointillist distribution because of the gappiness where it grows well. So you'll see little red spots that decay, and start growing elsewhere as gaps open up and close. Oops, let's go back. Okay, here we go. Very different dynamics from the preceding two species. Lots of gappiness towards the end of the run here. A lot of blowdowns. Now I'm going to show you a canopy tree species, Schmedia. Again, responding to a big blowdown here. It had a lot of recruitment early on, but not near, it's shade tolerant, so it doesn't have nearly the response that Hybanthus had to the early mortality of the canopy layer. But it still shows strong dynamics. So this is, I can guarantee that if you want to see the rest of these, they look pretty much the same. Although the individual patterns are different, of course, the dynamics are equally strange. So if we make a static look at these patterns, again, the same thing. Here we have intrinsic rate. This is Trichilia, the most common canopy species, in 2 census intervals separated by 15 years. And my point here is to show how different the pattern of population growth is after just 15 years of lag. So red is again sites of rapid population growth, and blue and purple are sites of rapid population decline. Here's another species, mid-story species, showing again the change in the pattern, and an understory species down here. So I call this boiling mud pot population dynamics. And I'm thinking of, if you go to Yellowstone and see the mud pots there, it reminds me of little patches of explosions of gas coming up, and no regulation at all, or very poor regulation at the local scale. And it's not clear that it occurs at the large scale either. So what's explaining these spatial patterns in population dynamics? Well, this is where we come to the end of the talk and the idea of the enemy susceptibility hypothesis, for which we think we have mounting evidence. And the importance of this is that this is not a regulatory mechanism. This is a destabilizing mechanism because you are killing rare species, not promoting them. They don't have an advantage. So what is this hypothesis? Well, here are the elements of it. A major cause of BCI tree death is structural damage caused by heart rot and root rot fungi. The boiling mud pot dynamics, according to the hypothesis, are due to spatially and temporally lagged predator-prey dynamics. Basically, the idea is that trees disperse and local stands of a given host tree species increase in abundance before fungal pathogens discover them. In other words, this is the lag. But once infected, the trees manage to live for years, continuing to reproduce, and they die of the infection, but it takes a long time. This increases the lag. So here's the hypothesis. Most tropical tree species are rare because they are more susceptible to heart rot fungi, or show more or share more fungi with other tree host species than do the common species. Therefore, rare species have fewer, quote, safe sites, unquote, in which to regenerate than common species do, because there are fewer places for them to grow that are far from a source of fungal infection or fungal contagion. So one of the predictions of this hypothesis is that rare species should become hollow-trunked at smaller diameters and at younger ages. So we can test this idea. And so here's one of our Panamanian researchers working on this question. Here's a tree infected by heart rot and butt rot. Roots get infected by hyphae crawling from neighbor infected trees. Then those hyphae grow up into the butt of the tree, infecting it and decomposing the cellulose of the heartwood. And then they invade higher and take out the heartwood itself. And this causes a structural instability and the thing snaps off or tips up and uproots. So one of the cool new instruments that we have is a portable CAT scan machine. And we strap it on the trunk and it takes a cross-sectional picture of the tree. And if it has a hollow in the center, we get this purple blotch in the middle that tells us where the heart rot is. If we find heart rot, we core the tree, so we don't have to cut the tree down to find that it's infected. In fact, we can wait and see if its mortality is accelerated by this rot. But we then can sample the fungus and culture it, get it growing in pure culture, identify it, sequence its genome, and then test it against a whole variety of species to see how broad its host range is among the different trees. So basically, so far we have tested 1,700 and some living trees this way, 173 species in 49 families, and we're looking at larger trees which are more likely to be infected, 20 to 131 centimeters. Here's where we've sampled the plot. And here are the three mechanisms. The expectation is that as trees get older and bigger, they are more likely to have heart rot. The classical view was that heart rot would increase with abundance of species, but the enemy susceptibility predicts the opposite, that the rare species will be more infected than the common species. So what do we find? Well, first of all, we find that large trees indeed have a higher probability of heart rot or decay in them as a function of diameter. So that's good. And secondly, we find that the number of species with less than 2% heart rot is a fair substantial number of them, but there are lots of species that have much greater levels of heart rot. And then the question, most important of all, is whether rare species are more likely to have heart rot. And the answer is yes. This is the plant abundance down here, over 20 centimeter trees. And the rare end is on this end. You see these curves are all higher here than they are over here. We're fitting logistic regression here. So we have zeros and ones, dead, alive. And you can see that the number with 2% decay is highest for rare than common. The number with 10% decay is higher rare than common. And the number with 50% decay is higher with rare than common. So we're getting there. We're very excited about our findings. So what we now have is a large greenhouse with 100 different species growing saplings. With the axenically pure cultures, we make infected toothpicks, which we of the word of the species, and we insert this in the stem of the tree, and we watch and see whether the hyphae grow into the heartwood of the host. So this is how we're measuring host specificity. And the prediction is that common species will be more resistant to more of the pathogens than the rare species, but we're in the thick of this and we don't have the answer to that question yet. So here are the take-home messages again. It's not an equilibrium. We have both chance and deterministic effects governing abundance. And there's evidence that rare species are more in trouble from pathogens. And this is a destabilizing mechanism that could explain why chance and determinism play such dual important roles in this particular forest. So now I pose the question of what's going on in paint rock? We don't know yet. Thanks a lot for your attention.
You did a good job.
I did?
Yeah.
Okay, well, I hope they liked it.
Right, there we go. Awesome. That was incredible. That was absolutely incredible. I've got pictures on my phone of all your graphics.
Well, I'm one of these people who tends to talk forever, so I apologize.
Well, it was perfect.
It was perfect. Now I'm thinking about gap phase dynamics and susceptibility, open cup nester susceptibility to cowbirds.
Yeah.
But then thinking about scaling, you know, and bird plots on top of these intensive vegetation plots, but at what scale we'll be able to measure or what's going on. So looking at nest predation, for example, as a factor, nest predation or brood parasitism, you know, open cup nesters and canopy nesters, which, I mean, it's hard to measure some of this stuff, but it's just incredible. These questions that started piling in, Steve. So thank you for that great lecture.
I'm glad you liked it, but I mean, you know, one of the things that's nice about having 40 years of data is 40 years of data. And, you know, I feel grateful to all the hundreds of people who've been working on BCI and their passion for this. And I hope we can keep this level of passion going in paint rock because, you know, it is the most diverse temperate forest probably in the world, according to Bill. And I believe it. I mean, this is just, we're likely to get up to how many species, Bill? Maybe 120?
Wow.
Well, I would caveat the most diverse temperate forest with a couple of things. Maybe the most diverse deciduous temperate forest.
I think I need
It's
to
hardwood
play the
heaven.
camera.
Yeah, hardwood heaven. So we, you know, we're well over, I think we're well over 75 species now. We've got a lot. The problem is, of course, we've got these new or unidentified species, I'll call them. And we have quite a few of them that are clearly not fitting in any category. So we could see more than 100 species of woodies on the plot before we finish. We potentially could see up to 120, but I would say somewhere between 100 and 120.
Well, that's still a very large number. You know,
Yeah.
one of my plots was in Costa Rica in dry forest, and we had 112 species. So,
Yeah, wow. I didn't realize that. So that's very interesting.
Look at this.
You know, one of the things I'm thinking about, you know, I was thinking about your greenhouse experiments and your infected toothpicks, and then I began to think about woodpeckers as pathogen carriers and
pathogens, yeah.
and wondering if are there any studies, Steve, going on with vertebrates on top of vegetative plots in any of these places?
It's not nearly as consistent as the tree work, but the idea is, yes, there's been a very long-term study of mammal populations on Barrow, Colorado Island, and you know, really good data on the dispersal of seeds, secondary dispersal of seeds by mammals. But it needs a huge amount more attention. You know, the trees wonderfully are low-hanging fruit, as it were, because one of the things, you know, I'm a turncoat zoologist. I got a degree in zoology, but I wanted to do population dynamics, and it seemed like the animal ecologists I knew were stuck on how many there were. And I wanted to be able to know that instantly or soon and get on with my more, you know, other questions. But we still have to know what's going on with animals. And I think we need more creative ways to do this. There's the remote sensing methodology. They put trackers on jaguars and things like that and have watched where trackers of a jaguar meet an agouti, and one of the monitors stops moving. So I mean, but it's difficult work. So I'm open to any ideas you have.
Yeah, looking today, by the way, is the Kickstarter for Terra, that remote sensing, the MODIS for bird song identification. So Cellular Technologies drops that today, and it goes online. So we're thinking about trying to get Terra in Paint Rock somehow, and they're relatively inexpensive. So we just got to do more investigating to find out what that sampling methodology is like, because it's brand new. I mean, it's essentially being betaed now, but exciting stuff, exciting opportunities.
Yeah, a sidebar we'll talk about, Steve and Drew. We did get funding through Riverview for a MODIS sampling station, I think, and I just don't know what happened to that. So we got to figure that one out.
Cool.
There's a group in Germany, you probably know, what's his name? It does Move Bank.
I know his name, just a minute.
Anyway, they have, tracking devices on all these migratory birds in Europe coming up from Africa, and they can watch them. Look up Move Bank, and you'll see incredible stuff that they're doing that I think we might try to get involved with, because they'll want to, help us with. They've got a lot of funding from the German government.
Let me respond quickly
Martin
to
Wachowski.
Martin Wachowski.
Wachowski.
Sorry.
A question that Dr. Stone asked, and for the students, it's an important question, and Steve and I have talked a lot about it, and it's that really important functional question of what is a species, and where are we going to put the limits on species. Hybridization is occurring. Let's take oaks, and just, we have a pretty simple-minded idea of what hybridization means, but of course modern research is showing that that hybridization is far more complex than we usually think about. So there is a concept, and we're working with Andrew Hipp on specifically some of the oaks in our plot which have been problematic. And that concept is I'm going to try to pronounce it right, Singaminon complex, which means that there is gene exchange between species, but the species pretty much retain their reproductive isolation and their characteristics. So we do see that, but clearly hybridization doesn't explain most of what we're seeing in paint rock in terms of not known species. So just for an example, we have this little paint rock woodmint that is really only known from paint rock. It's really cool. If Patience and Helen haven't shown it to you yet, they should. Shame on them. And
is that the Blophilia?
That's the Blophilia. And so Blophilia subnuda, it's also blooming at the same time as Blophilia hirsuta, which is all along the roads. But for a long time, people said, oh, it's just a hybrid. Well, no, it wasn't. And so there is a danger in referencing hybrids. Hybrids have hidden a lot of species diversity, and we've got a rhododendron on the top.
You pointed out.
We've got a rhododendron on the top of the plot that for years was written off as a hybrid. Well, one of the big problems with it being a hybrid is there were no parents. It was up there by itself, like nothing else, and with nothing else that could have contributed to its genetics. And now we're probably going to have to give a new species designation to it. So hybridization is occurring, and we there's a couple of things out there that may be the result of hybridization, but that we're looking at now. But we think it's, there's just a lot of species that for whatever reason, we never really characterized because
I
the
have
people,
a question about this.
yeah,
What we see in Panama is very low hybridization rates. And I wonder whether the glaciation of North America isolated populations incompletely. And so there's much more hybridization in the temperate zone than in the tropics. I mean, that's my expectation. You want to comment on
yes.
that?
Yes, but of course that hybridization is if that's your theory, I would suggest that hybridization occurs very slowly and only in certain fringe populations, if you see what I'm saying. So
Yeah.
it's not widely distributed, but narrowly distributed. And I think that is occurring. And I think it's actually maybe resulting in some types of speciation as well. But yeah, so we got a lot of questions to ask there. But it's hybridization is important and we need to be aware of it and look for it. But we should also be careful about writing things off to hybridization as well. And Steve, I ask you this question a lot, and I don't know that I ever ask it the right way. I'm going to try it again. Do you think there's more edaphic variation on the paint rock site than there is in, say, Barrow, Colorado?
I do, because we specifically chose sites that had strong edaphic variation. So if you look around the Forest Geo network, plots that were purposely set up on discontinuities in soil, like the Lambier plot, for example, which has about a 70% turnover in flora within about 10, 15 meters. It's clear that if you choose your sites to have strong gradients in soils, that you'll see a lot more turnover. So we picked the BCI site originally because we wanted it to be as homogeneous as possible. So that's a problem for generalizing from BCI, because other plots were picked for different reasons. And so what I expect is that as you increase the gradient of differentiation of soils, you will start to see species unable to bridge adaptively those soil types. And you'll start to get a differentiation of species by local, what do you call it, pair patry, into separate populations. And there's a big soil discontinuity in Western Amazonia caused by, west of the Andes, or east of the Andes, there was a huge lake in the Miocene, because the uplift of the Andes was forcing what had been drainage to the Pacific to the Atlantic. And so in the intervening millions of years, there was a big lake with a very rich deposit of benthic, debris. And then when the Andes lifted more, you got rapid water streams that were nutrient poor on top of this. And so you have these areas of very rich soil interspersed with very poor nutrient soil, and a very strong turnover in flora. So I think that's another major question, is when do you disrupt the cohesion of a genome to being able to grow on all these different soil types? And BCI is clearly on the side where we have less differentiation of soils.
Yeah, and spatial questions become really more important in those kinds of studies. Am I correct? Yeah.
Yeah.
I had a question about the concept, the statistic associated with it was Fisher's K. It was the distribution of like rarity in that curve. I was just going to ask how what would a different curve look like in terms of actual species composition? Is there variation?
Yeah, that's a very good question. I can send you a copy of my book where I describe it, but I think it'd be simpler just to tell you that the curves vary quite a bit in steepness. So for example, if you go to a boreal forest with maybe 5 species, you get a Fisher's, it's actually Fisher's alpha. Fisher's alpha is a very small number, like 0.5. Whereas if you go to Amazonia, it's like 700. And if you go to BCI, it's 50. So the number varies, and the steepness of the curve that you get is controlled by that number. So you have more dominance when you have a small number and fewer species, and you have less dominance, like in a tropical forest, when you have a large Fisher's alpha and many rare species, and less concentration in the most abundant. So yeah, I mean, if you send me an email, I can send you a PDF of my book, but I can also include another couple of papers that are short reads that talk about this.
Yeah,